Good morning to Workshop 03 at the North American Cystic Fibrosis Conference in Seattle. Couple of logistics things. Feel free to have some breakfast. We are thinking that the room is probably going to fill up completely, so I was asked that everybody takes their bag down from a chair to 3 chairs, but also don't forget anything later. And then we have restrooms on both hallways on the side, similar distance. So I'm really glad to be able to welcome you this morning to a very interesting workshop that focuses on cell-specific roles of CFTR in the airways and insights into expression and function. I am co-chairing this workshop with the wonderful John Brewington from Cincinnati Children's Hospital, and I'm Martina Gensch from the University of North Carolina at Chapel Hill. We have a fantastic mixed program that will start off with looking into ways of detecting CFTR and then assessing which cells are actually important in the airway epithelium for CFTR expression. We will then hear about the role of CFTR in alveolar type 1 cells and also discuss the role of CFTR in ionocytes and finally end with the role of airway glandular myoepithelial cells in regeneration of high CFTR expressing cells. And it's now my big pleasure to welcome Victoria Moimer from the Cystic Fibrosis Foundation Labs, and she will speak on the generation of a robust CFTR-specific immunolabeling protocol across cell cultures and tissues. Victoria.
Good morning, everyone. My name is Victoria Moemeyer. I'm from the CFF Therapeutics Lab in Lexington, Massachusetts. And today I'm very excited to share with you work that's been done in collaboration with University of North Carolina to generate a robust CFTR-specific immunolabeling protocol across cell cultures and tissues. I have nothing to disclose. So in the realm of CF research, gene therapies have come a long way, but there's still lingering questions. What is the ideal cell target for CF therapeutics? And how can we standardize the way in which we measure therapeutic success? We believe that by defining where CFTR is expressed, we can actually identify that target cell type along the proximal distal axis of the human airway, as well as directly measure success of potential therapeutics by actually visualizing CFTR restoration. This ultimately led us to develop a CFTR immunostaining protocol that's highly specific, highly sensitive, and adaptable across several model systems to achieve all of these goals. We began our optimization process in the simple cell landscape of 16 HBEs. Now, the rigorous optimization went into this, but our goal readout for initial optimization was genotype specificity. In other words, having specific signal in the non-CF line and no signal detected in the CF line. And you can see here that we were ultimately able to achieve that result after a long time of optimizing this protocol. And you can also see there's very good signal to noise ratio here with very little background. To achieve this result here, we first began with a 1.6% paraformaldehyde fixation, followed by a permeabilization with saponin. It's important to note that this differs drastically from the typical histological process of 4% PFA, followed by Triton-X 100 permeabilization. But we found that the combination of these two components here is critical for preserving the CFTR signal. Other reagents may completely ablate the signal. Other reagents may bring up a lot of background. So these two steps are key. We follow this with an incubation with UNC 596, which is specific to the NBD2 domain of CFTR. The work that we did was optimized mainly on this antibody, and the majority of the data I'm going to show you today is using this antibody. However, we have been able to demonstrate reproducibility of this protocol with UNC 528 and UNC 450, which are specific to different epitopes of CFTR. Our protocol then follows an HRP conjugated secondary antibody incubation and a tightly time-controlled tyramide signal amplification to ultimately achieve the result you see on this slide. So Now that we have shown that we have a protocol that is genotype specific in 16 HBEs, we wanted to explore how sensitive this protocol was and start to get into what is the lower level limit of detection of this protocol. Now, there's a lot of data on this slide, so I'm going to walk you through it. But essentially what we aim to do here is slowly titer up the amount of CFTR protein in 3 different PTC variants in 16 HBEs. By doing so, we hope to run this alongside a matched Western blot to get a semi-quantitative readout that hopefully would show a matching of CFTR IF intensity and band intensity of band B and band C on the associated Western blot. So to begin, no treatment in these 3 different lines, G542X, R1162X, W1282X. You see no signal in either the immunofluorescent landscape or the Western blot landscape. Similarly, with CC9K9, which is an ERF13 degrader, you don't see signal in either realm as well. Now, it's difficult to see on this slide. I apologize, the lights are washing it out a bit, but you could see it much more clearly on my poster 300. But there is faint IF staining in the W1282X ELOC02 treatment, which is another read-through agent. And similarly, you can see faint band B and band C for that condition. And finally, when we combine the 2 treatments, you can see very clear CFTR staining in the W1282X, very strong banding pattern for that same condition in the Western blot, as well as, again, faint CFTR signal in the R1162X dual treated, with faint banding in the Western blot. This Western was run alongside a 2% wild type control. And ultimately what this large data set showed us was 2 things. That the intensity of the CFTR IF stain that was brought out from our protocol aligns very closely with the Western blot, and thus the amount of protein that's present in that sample. And our protocol can detect down to at least 2% of wild type signal. Now that we were confident we had a specific and sensitive protocol in a simple cell landscape, we felt ready to move into a more primary cell landscape, human bronchial epithelial air-liquid interface cultures. As with the 16 HBEs, we first wanted to confirm genotype specificity. And you can see here that we were able to do so. We have a non-CF culture that was stained alongside a CF culture. In this case, it's a dual PTC, G542X, R553X. You can see CFTR staining across the culture showing up as strong spots of signal with some weaker signal interspersed and complete absence of signal in the CF sample. But now that we're working in an ALI landscape, primary cell material, these are mature, differentiated cells and different cell types are represented. So we can start to ask questions like what cell type is making the signal that we see here? And we can see those strong signal spots are associating pretty much exactly with the apical domain of BSND positive ionocytes. Although you find those ionocytes in the CF material, they are not expressing CFTR. This pattern of expression was replicated in a protocol that was developed in parallel by our collaborators at UNC, shown on the right. You can see once again that that strong signal is replicated across the culture. It's apical, and it does not associate with multiciliated cells at all. And once again, we can see that that strong signal is also associating with the apical domain of ionocytes. So while we've shown that we can detect native CFTR across different protocols with genotype specificity in HB-ALIs, we wanted to explore a more therapeutically relevant application of our protocol. So as I mentioned at the beginning, delivery is a pretty hot topic in CF research today, especially what is the ideal vehicle for getting CF therapeutics to the appropriate cell type in the human body. Some of the most looked at vehicles are lipid nanoparticles and viral vectors. So we wanted to test the ability of our protocol to detect not only native CFTR, but also ectopically expressed CFTR that has been delivered to a CF culture by either LNPs or lentiviruses. So the 2 different experiments I've described to you are on the top and the bottom. They are unrelated experiments, so they shouldn't be compared side by side. But what you can see in both cases is that using our staining protocol, prior to treatment in either case, you see no CF signal in the CF HBE ALIs. However, after either LNP or viral treatment with CFTR, you can very clearly see CFTR signal showing up in those cultures. So we've confirmed that not only can our protocol detect native genotype-specific CFTR in primary HBEs, it can also detect ectopically expressed CFTR, showing that it has future implications for in vitro readouts. Finally, we can start to understand what is the actual CFTR expression in the context of the human airway. So once again, we looked at genotype specificity. For all this data I'm going to show you for the next few slides, this is all in the formalin-fixed paraffin embedded landscape, so just keep that in mind. In the non-CF control, we found two distinct patterns of CFTR expression. The most frequently observed pattern was the one in the top left. That's our apical expression pattern. That's what we most frequently saw. But we also saw this rare pattern seen in the top right, which we'll call pericellular. That was a very rare pattern that we found. Notably, you'll see that there is no CFTR signal in the CF negative control material, which is also G542X, R553X. Now, I am showing you genotype specificity, but with the FFPE landscape, there's other variables you have to account for and that are difficult to control for, such as differing ischemic times, differing fixation times, and limited availability of CF control material. So we wanted to make sure the study was appropriately powered. We replicated that wild-type pattern across 9 different non-CF donors just to support that this was a reproducible pattern and that this protocol is robust as well as genotype specific. So finally, we're at the big question. What cell types are expressing CFTR in the human airway, and is it differing across the proximal distal axis? We wanted to begin by looking in a non-CF airway at our major cell types, our CC10 positive secretory cells, our BSND positive ionocytes, our multiciliated cells as marked by alpha tubulin, and our K5 positive basal cells. When we stain those with CFTR, we can see apical expression of CFTR in both CC10 positive secretory cells and ionocytes. That's where we most frequently found the CFTR signal. We did see this rare pattern in the K5 landscape here, where you can see CFTR on the apical surface of what we believe to be a suprabasal cell that is extending towards the apical surface of that airway and differentiating into another cell type. This expression pattern does align with what we observe for those transitioning basal cells in single-cell sequencing data. So now that we looked at our major cell types, we want to look at the entire context of the human airway to really understand where we need to be targeting. So first and foremost, it's important to understand the airway is not uniform as you move proximal to distal. In fact, a paper out of the Tata lab in 2022 showed that you could separate the airway into distinct zones moving proximally to distally based exclusively on the composition of the secretory cell population in that particular airway. For example, you only find MUC5AC positive goblet cells in the most proximal of airways, whereas as you move down distally, you don't see those cell types as much anymore until they completely disappear. And then you start to see other secretory subtypes such as SPB positive and SCGB3A2 positive cells in our most distal of airways. So we first wanted to identify, across serial section analysis of a non-CF lung, can we identify airways from each of these distinct spatial zones? And using the same markers shown in the figure in the middle, we were able to do so. So we have 3 airways representing 3 distinct regions of the human airway trait. So now that we've done that, we wanted to ask the question of, does CFTR expression differ in these regions? And what we found was that when the different secretory cells were present in their respective regions, they were all expressing CFTR. Now, not every member of every secretory cell subtype was expressing CFTR, but I'm showing you instances in which I was able to identify apical co-expression. But you can see that the overall pattern of CFTR is sort of that apical expression in all 3 of these regions. And although the secretory cell subtype expressing the CFTR may be different, the overall pattern seems to be pretty similar, moving from proximal to distal. So overall, what I've been able to show you today is that we have a protocol that is highly sensitive and highly specific for immunostaining of CFTR across multiple different landscapes, including cell cultures and tissues. And this protocol is capable of identifying native, read-through, and ectopically expressed CFTR. When we look at the expression pattern of CFTR in the human airway, while it doesn't differ too much in terms of overall expression pattern, moving proximal to distal, and we mostly see it in secretory cells and ionocytes, that subtype of secretory cell may differ depending on where you are in the airway. As we look forward to more therapeutically relevant applications of this protocol, our collaborators at UNC were able to generate this very new data set, which is using non-CF human bronchial brushings. You can see using the staining protocol that we have shown you in this slide deck, they were able to identify apical CFTR expressing in both ionocytes and secretory cells. So as we're looking for ways to standardize therapeutic success in the clinic, this has pretty big implications for how CFTR immunofluorescence can be used to directly measure that therapeutic success. So there's a lot of people to thank here, the entire CFFT lab shown in that image here, but in particular, my team members, Junji Liu, Sam Raskin, Jake Lesser, Alex Stufer, my advisor, Jed Mahoney, as well as John Carulli and Martin Mentz for their guidance and support. Of course, our collaborators at UNC, Martina Gensch, Debbie Shalon, Tim Jensen from the antibody program at UNC, who helped us get these antibodies that work beautifully in our protocols, and Andrew Gio from the US EPA, who aided in acquiring the bronchial brushings that you saw in the last slide. And I will take any questions. Thank you.
We take questions from the audience and also online. Feel free to ask.
That was a great talk. I'm wondering if you can comment if and where you saw CFTR in the glands.
Yes, so we have done some CFTR staining in the submucosal glands, and for the most part, it's this apical expression sort of along the inside layer of the SMGs. But yes, we do see it there.
Mucus or serous cells or both?
We haven't done a ton of analysis into the different cell subtypes there, just that we can see it in what we identify as submucosal glands. So that's something that we're looking to do in the future.
Awesome, thank you.
I have a similar question about the airway parenchyma, AT2 cells, AT1 cells, any CFTR there?
Sure. So we have looked a bit at the alveolar space, identifying AT2s and trying to see if it co-expresses with CFTR. Thus far, we haven't seen much. I know that there is data that does show that in terms of RNA expression, it's expected there. So at this point, we're not sure why that is, but that is something that we're looking to explore in future studies.
Yeah, that's interesting because if you look at the single cell data, AT2 cell expression of CFTR really is pretty high. So there seems to be some kind of disconnect then between.
Yeah. I know we were kind of surprised by that result as well.
Yeah.
You know, we are looking to do some more RNA studies as well as like RNA scope, RNA in situ hybridization along with the CFTR immunofluorescence. There is some data for that on the poster as well, but there's some interesting observations with that we're not entirely sure how to explain at this point.
And then I also noticed in your cell culture data, you could clearly see the ionocytes, but could you also see the secretory cell expression of CFTR in that cell culture?
Yeah, so it's much, much dimmer, but it is there, and it's probably not showing up as well on the screen here.
All right.
But we just wanted to show that, you know, that bright signal was ionocytes. But yeah, we do see it in the secretory cells.
Thank you.
Mm-hmm.
Yeah, great talk. So I come from an LNP lab, so I want to ask you about the LNP delivery in the differentiated like CF patient cells. So based on the IF staining, do you have an estimation of how much protein level rescue was like can be observed based on that?
I admittedly don't know a ton about the LNP experiments that were done, the ins and outs of them. I would say just by looking at the cultures, you know, it wasn't across the entire culture, if that's what you're asking. I sort of saw, at least for this particular experiment, patches of expression in different areas of that culture. So it wasn't uniform. In terms of percentage of the total culture that was hit with the LNP, is that what you're asking?
Like in total, how much protein function rescue, like,
In terms of protein function rescue, that I don't know off the top of my head, but you can ask anyone in the CFFT lab that's probably in this room, and they could help you more with that than I could.
Thank you.
Last question.
Hi, I was just curious to know what you think the identity is of the cell type that has the paracellular CFTR expression.
We feel pretty confident at this point that that is an ionocyte.
Okay, great. Thanks.
Let's thank Victoria again.
Excellent. So now that we've identified our ways of finding CFTR in the airways, happy to have Dr. Carlos Farinha, came all the way from Lisbon, to come up and share with us. Let's see. Gotcha. He is going to be thinking about what cells do we need to target in the airway epithelium. Thanks, Dr. Farinha.
Thank you very much, John. So, morning, everyone. I'd like to start by thanking the moderators for selection our work, in which we are developing a strategy to try to identify which cells to target in the airway epithelium using adenine base editing. And I start by saying that this is part of an ongoing collaboration between my lab in Lisbon and Patrick Harrison's lab in Cincinnati. So these are, I have no disclosures related to this presentation. We are all pretty much aware that the approval of triple combinations for CF has provided a causal therapy to most people with CF, around 90%, if we look at the numbers recently published. But we know that modulators do not target mutations that abolish protein synthesis. So gene-based therapies are being pursued because they represent the best possibility for a cure for these people with CF. And among these gene-based therapies, there are the gene editing approaches that include homology-directed repair, and more recently, the prime editing and the base editing, specifically the adenine base editing approaches that can correct specific mutations. And the adenine base editing approach uses a Cas9 niacase fused to an adenosine deaminase enzyme that can convert any adenine in the editing window into a guanine. And the ABE system is double-strand break-free, so it has the possibility of introducing changes and significantly reducing the indel formation. And interestingly, base editing can correct not only the adenine, but also taking into consideration the other base editors that have been developed, can correct around 40% of CF causing variants, being adenine base editing the most useful approach because it can correct around 25 to 30% of CF causing variants. And so here in this study, we focus specifically on the ability to correct W1282X, well known as the second most common nonsense variant in the CFTR gene, and that is not eligible for the currently available modulators. And in fact, we have previously shown that using homology-directed repair that we can correct W1282X. We observed in 16HB cells around 20% of correction at the DNA level that corresponded then to a recovery in a rescue of wild-type CFTR at around 10% of expression normalized to wild type and around 20% of CFTR function. But however, for each allele bearing W1282X that we were able to correct, around the same number of alleles was disrupted by the creation of indels created by non-homologous end joining. We have also used Patrick's lab with our collaboration and collaboration with other people in the field. We have also used adenine base editing to correct W1282 W02X, so to avoid the formation of indels and to correct the A back to a G to rescue this mutation. And in fact, using HEC293 cells, the Philippine version, and the guide RNA targeting the adenine at position 6 and NGAB max base editor, we were able to detect around 25% of correction in the amplicons analyzed. However, one of the problems with adenine base editing is the introduction of bystander effects. So if you have other adenine residues in the editing window of the base editor, this can also be corrected. And in fact, if we look to our results in the HEC293 cells that pretty much are recapitulated in primary human nasal cells and also human bronchial epithelial cells. For each allele that is in which the correct adenine is edited, more or less the same amount is also changed in an adjacent adenine. And so this raises the question that an improved adenine base editing tool needs to be developed. So to do this, we started by creating a suitable cell model, and we used the BCI cell model. So it's a basal cell line that retains the ability to differentiate into the different cell types in the airway epithelium. And so using homology directed repair, we created a model based on the BCI wild type cell bearing W1282X in homozygosity. So we were able to introduce the variant, as you can see here, confirmed by Sanger sequencing. And then in this new model that we created, we cultured the cell in air-liquid interface for 30 days, and we assessed CFTR expression at both RNA and protein levels. And we can see, as expected, that there is a reduction in the levels of CFTR RNA, most likely due to nonsense-mediated decay, and almost no detection of CFTR protein. Then we also confirmed in this newly created cell model that it still retained the ability to differentiate into the different cell types. So we cultured the cells in ELI for 30 days, and then we assessed markers for basal cells, keratin 5, ciliated cells, FOXJ1, goblet cells, MUC5AC, ionocytes, FOXI1. And we detected the levels of these markers, and we saw no significant changes in the levels of these markers in the novel mutant cell line compared to its wild-type counterpart, so confirming that the new cell line retains the ability to differentiate into the different cell types. And then we tested adenine base editing in this cell line. And so using this time an SPRY AB 7.10, which has a similar editing window as the one that I previously mentioned, we assessed editing in this cell line. And once again, we observed around 5% of amplicons with the correct editing, 2% with just the neighbor adenine rescued and then, or changed, and then also 5% with the two adenines change. And so this confirms that adenine-based editing is able to introduce the correct change in the DNA, but it has, with minimal indels, but it has significant bystander defects. So confirming that an improved ABE tool is needed. And that's why we focused on developing a strategy to reduce unwanted edits, so to reduce off-target and bystander effects, by having temporal spatial control of the editing. And so we focused first on the temporal control of the editing. Try to edit when we want and not just because we transfect the cells with the adenine-based editor. And so for this, we based ourselves in a previous work describing a split version of Cas9, and we built a split version of the adenine-based editor of the SPRY. And so in this construct, the C-terminal fragment of Cas9 is fused with FKBP domain, and the N-terminus fragment of Cas9 with the adenine-based editor are fused to the FRB domain. And so, as you may know, FKBP and the FRB are mTOR domains that only assemble in the presence of rapamycin. So we developed a way of making the editing only when we treat cells, transfected cells, with rapamycin. This construct also contains a U6 promoter-driven guide RNA, a beta-actin promoter, nuclear localization. Well, in fact, we have two constructs with either with both nuclear localization and nuclear export signals, or only nuclear localization signals in the C-terminal fragment of Cas9, and also 2A self-cleaving peptides to separate the two halves of Cas9. Then we tested this construct in this novel cell line, so the BCI W1282X cell line, that we transfected with a plasmid including either the split ABE or the intact ABE, and we treated the cells with either rapamycin or DMSO as vehicle control. And so we can observe some editing even without treatment with rapamycin, so under control conditions. So there is some spontaneous dimerization of the adenine base editor even without rapamycin. We also see a significant increase in the levels of editing when we treat the cells, about threefold increase when we treat the cells with rapamycin, and interestingly, almost no bystander effects. So we are still continue to characterize this split ABE system and to try to understand if these low levels of bystander effects are just due to the fact that the editing, the correct editing, is also on the low side. And we are trying to improve the guide RNA that we are using. And so for this, we performed a guide RNA tiling assay in egg cells to introduce some optimization to increase specific editing and decrease the bystander effect. And in fact, the results that we are obtaining from this show that if we target adenine at position 5 is better than targeting, is more efficient than targeting the adenine at position 6. We observed doing this transfection of the construct in HEX cells, so these are not the BCIs. We observed on-demand control of the editing of the split ABE, the one here in dark blue, with 5-fold increase compared to the control conditions. We also observed that the split ABE has a very similar editing window to the intact ABE, and we observed minimal spontaneous dimerization of split ABE and low levels of editing, even in the absence of rapamycin. If you want to know more specifically about these improvements of the split ABE tool, please visit Joana Alves at poster 275. And as we are continuing to optimize the split ABE tool, we are also focusing on the second part of the strategy, on the spatial control to try to edit where we want. And so for this is quite important, as we have already heard today. It is not clear which cells and to which amount need to be edited in the lung epithelium to restore CFTR functions to levels that correspond to clinical benefit. And so for this, we developed a strategy to try to express our split ABE tool in specific cell types. So, in which the split ABE construct is being cloned into lentiviral vectors with promoters specific for the different cell types. And we did this by sequential cloning in a series of 3 steps. The first one, we started with a lentiviral vector in which we replaced the puromycin resistance gene by the anigromycin resistance gene, because the BCI cell lines that we are using are already resistant to puromycin. In the second step, so we use this created lentiviral vector with agromycin resistance, and we cloned our split ABE construct with an appropriate spacer using two unique restriction sites. And then in the third step, using these 2 unique restriction sites, we replaced the beta-actin promoter with promoters specific for the different cell types. So keratin-5 for basal cells, FOXJ1 for ciliated cells, MUC5AC for goblet cells, CC16 for club cells, and FOXI1 for ionocytes. And the other promoters are also in the run to have more than one promoter specific for some of the cell types. And so with this, we are creating a new CRISPR-based tool for spatial control of the editing in specific cell types. And so we then started by confirming that the constructs can be expressed in the BCI cells. So we transfected the cells with a lentiviral vector. So this is a transient transfection of the lentiviral vector, in this case, the one with the keratin 5 promoter into the BCI cells. And then we treated the cells either with a control or rapamycin, and we assessed the expression of Cas9 by Western blot. And we can see that Cas9 is expressed either with or without rapamycin, because rapamycin will induce the dimerization. And also assess the localization of, and we detected the expression of the two fragments, so the C-terminal and the N-terminal fragment. And we also assessed the localization of the two halves of Cas9 by immunofluorescence. And we can see that the C-terminal fragment doesn't change that much its localization with the treatment of rapamycin, and it's mainly localized in the nucleus, whereas as expected, the N-terminal fragment, when we treat the cells with rapamycin, shifts from the cytosol to the nucleus because it's expectedly it's dimerizing with the Cas9 C-terminal fragment. We then transfected packaging HEC293 T cells to produce lentiviral particles, and after quantification of the particles by the digital drop PCR, we transduced the novel BCI W03 cell lines with different MOIs, and they are now under selection with hygromycin. We are also working on an alternative strategy for cell line development, so to better assess the packaging, and also to be able to select the cells that are stably transduced. We also produced alternative versions in which the hygromycin resistance gene has been replaced by turbo GFP, and so we have now the full set of constructs in which we have turbo GFP with the different types of promoters. We are also assessing the expression of these vectors in the BCI cell line. So as I've shown before, in this case we electroporate the cells with either the hygromycin construct or the GFP construct. 24 hours after transfection we treated the cells with DMSO or rapamycin, and then we assessed the levels of Cas9. In this case, I'm showing the C-terminal fragment by either Western blot or immunofluorescence, and we can see that we have a pretty good expression. Seems to be even higher in the presence of rapamycin for the GFP construct, and some efficient transfection of the cells with this construct. So we are in the process of developing the novel cell lines. We have the tool that has been produced. So overall, I've showed you our ongoing process to develop a strategy for temporal spatial control of CRISPR-ABE to correct W1282X, with a long-term goal of understanding which cell types and of how many of them need to be corrected to restore CFTR function to therapeutic levels. Let me just end by acknowledging people who did the work in both our labs. So Tita in my lab, and Joanna, joint PhD student in Patrick's lab, and CFF for the funding, and thanking you for your attention. Thank you very much.
We take questions.
Carlos, very nice work, Carlos. I'm just thinking of why splitting the cast makes a difference. Have you considered coordinating the administration of the rifampicin with cell cycle, for example, or trying different concentrations of that?
That's one of the things that is on the go, to assess different times of treatment with rapamycin and different concentrations. Because our idea at this level is probably because we think that the reduction in the bystander effects is linked to the fact that the editing itself is not very high also. Mm-hmm. But hopefully, when we get the novel cell lines with so stable transduction, we will be able to treat for longer periods with different concentrations. So
Great.
Hi, very nice presentation. Thank you. I have a question regarding another class of cell types that, if you think, in your opinion, that should be targeted, which are the immune cells, and what could be the challenges in that case for targeting those the immune cells for the gene-based therapy?
Well, with the construct that we have, we can replace it's not that difficult. It's just a cloning step. We can replace the promoter with an different types of promoters for different types of immune cells, and we can test in the same way we are testing here. So it shouldn't be a big challenge to do that because it's a simple cloning step.
Yeah, but do you think it is important to target the CFTR defect in these immune cells? Because
well, that's another discussion, of course. I would say that in principle, if you go to the principles, in each cell in which you have CFTR, it would make sense to correct CFTR. Yeah. Just going to a philosophical discussion on the need of having CFTR. If CFTR is there, you would think that CFTR is important in every cell in which it is expressed.
A matter of time, let's move on to the next.
Hi, thanks, Carlos. I was curious, have you confirmed that your promoters are cell type specific?
We are in the process of doing that, yeah.
Great, thanks.
Yeah.
Eric?
We just to add a little bit more, we also tested, so the thing that I've shown you for keratin 5, we did the same for the FOXI one, and we see some leaky expression in a non-differentiated cell type. It is a non-differentiated, yeah.
Yeah, I mean, naively, I would expect that they might work really well for some genes and not as well for others,
Exactly.
based upon, how those genes are controlled, and really.
That's why we are also trying to have different promoters for the same cell type, yeah.
Yeah, makes sense. Thank you.
Carlos, lovely presentation. Could you comment on sort of the long-term plan, how you'll determine using this technology, which cells are the most important to
Yeah, so the long-term plan is, as we are able to add it when we treat the cells with rapamycin, is doing, after we optimize the time that we need of treatment with rapamycin and the concentration, is to treat with rapamycin at different points. So transduce the cells, differentiate the cells before and after being treated with rapamycin to check if we need to rescue just the basal cells, and they will be able, after being rescued, to differentiate into an epithelium with some levels of corrected function, or then add it when we have a stratified epithelium at air liquid interface.
Sort of transgenic animals and with the constructs. Thank you.
Excellent. Thank you very much.
And to Antonio and the other immunobiologists in the room, we selected for airway talks. So we're not ignoring the immune system, but we're focusing on the airway today. So we've thought about how do we identify CFTR. We've thought about what cells do we need to target. Now we're going to shift focus, and the next 3 talks will be focusing on CFTR function and some specific cell types in the airway. We're going to go distal to proximal. So excited to have Stephanie Tang come and talk with us from ICANN School of Medicine at Mount Sinai. She'll be talking about CFTR function in alveolar type 1 cells. So, Stephanie.
Hello, good morning, everyone. Thank you all for inviting me today to discuss our project in the lab. Oh, sorry. My name is Stephanie Tang. I'm a graduate student from ICANN School of Medicine at Mount Sinai in New York. Today, I'll be talking about our work in the lab that looks at CFTR function in the lungs and its role in alveolar defense against pathogens. I have no financial disclosures. Okay, so to get started, in our lab, we study lung infection that cause acute lung injury, which is a disease in lung alveoli, the main area of focus in our lab. So I'd like to direct your attention here to this cartoon of single alveolus. It's been well established that in alveoli, there is this liquid lining layer known as the alveolar wall liquid. This alveolar wall liquid is constantly secreted by CFTR function in the alveolar epithelium. The secretion generates a liquid flow across the alveolar surface that clears inhaled particles and bacteria out from the alveolar airspace. In our lab, we recently showed that when CFTR function is blocked, either by viral infection or CFTR drug inhibitor, the secretion is inhibited, which allowed staph bacteria to stabilize against the alveolar wall, establish its infection, and cause acute lung injury. Now, this is an important finding for our work because it showed that alveolar wall liquid secretion protects alveoli against bacterial infection. And so next, we wanted to know, well, what cell types in the alveolar epithelium drive alveolar liquid secretion? Evidence from published literature have shown that alveolar type 2 cells are important, but what about the alveolar type 1 cells? Since alveolar type 1 cells do express CFTR and they make up nearly all of the lung surface area, we consider the possibility that CFTR and alveolar type 1 cells contribute to alveolar wall liquid secretion and alveolar defense. So to test our hypothesis, we used existing transgenic mouse strains to generate a tamoxifen-inducible Cre-Lox transgenic mouse with CFTR deficiency and GFP expression, specifically in alveolar type 1 cells. In other words, after tamoxifen treatment, the Ager Cre-ERT will direct CFTR deletion to CFTR flock sites, specifically in alveolar type 1 cells. These transgenic mouse lines, they also have the ROSA MTMG allele that will direct GFP fluorescence to Cre-positive alveolar type 1 cells, which in the coming slides I will be showing with a cyan pseudocolor. And it will also direct TD tomato fluorescence to Cre-negative cells, which I will be showing with in magenta pseudocolor. And of course, for our littermate control mice, they will have all the transgenes except for Cre. So next, in order to visualize alveoli and the fluorescent Cre expression, we perform a mouse surgery where we put 3 cannulas, one in the pulmonary artery, one in the left atrium, and one in the trachea. We then remove the heart, lungs, and all the cannulas together as a unit, and we put it under a confocal microscope. We'll connect the trachea cannula to a ventilator to inflate the lungs, and then we'll connect the other 2 cannulas to a pump that will perfuse the lungs. Then at the end of this setup, we will have a live, intact, perfused mouse lung where we can view alveoli in real time. So this is an example image of what we see under the microscope. I will get into more details on the next slide, but briefly, you can see that the black areas are the alveolar air spaces. The cyan fluorescents are the Cre-expressing alveolar type 1 cells, and the magenta fluorescents are all the Cre-negative cells. So I'm going to go ahead and show you the cell type specificity of CFTR null expression in our live lung model. So this first image here shows a low power view of an alveolar field as you've seen in the previous slide. And these are from lungs of an alveolar type 1 CFTR null mouse. Again, I'm using the magenta pseudocolor to show the Cre-negative cells and the cyan pseudocolor to show the Cre-expressing cells. To better see the cell type specificity, this first high power image here shows Cre expression in the thin airspace facing alveolar type 1 cells, whereas the cells lining the adjacent vessels are Cre-negative or in magenta. This tells us that alveolar type 1 cells express GFP and are CFTR deficient. This next high power image here shows the cells that lack Cre expression and has taken up a dye that labels lamellar bodies, indicating to us that it is an alveolar type 2 cell. This also tells us that the alveolar type 2 cells, they don't express Cre and have CFTR intact. In the littermate control mice, we see no Cre expression as we'd expect. Finally, we also confirm cell-specific loss of CFTR RNA by doing qPCR. So this data, together with our imaging data, we interpret that CFTR deficiency is limited to alveolar type 1 cells. So next, we wanted to understand the alveolar liquid dynamics in these alveolar type 1 CFTR null mouse models. So again, we went back and used our live lung imaging approach to determine alveolar wall liquid secretion. So to do this, we micropuncture a single alveolus with a fluorescent dextran liquid tracer, which will allow the spread of the dextran tracer to a field of alveoli. Over time, we'll observe a time-dependent loss of dextran fluorescence, and this dextran stays within the alveolar airspace and it cannot cross the membrane barrier. So we'll interpret that dextran fluorescence loss signals dextran dilution by alveolar wall liquid secretion. To confirm our dilution assay, we did separate calibration experiments where we took glass micropipettes and diluted stock dextran concentration, and we showed that by diluting the stock, we could decrease the dextran fluorescence. Here at the bottom left is an example image of what we see under the microscope with the dextran. And you can see that it fills the alveolar air spaces and it pools in the alveolar corners as we'd expect. Now for each of our imaging experiments, we check for barrier integrity, which rules out the possibility of the dextran leaking out from the alveolar air spaces to ensure that the loss of dextran fluorescence we do see is due to alveolar wall liquid secretion. Now, I didn't have time to fit the data into my talk today, but we also ruled out the possibility that the micropuncture itself could induce some dextran fluorescence loss. So we nebulized the dextran and also found the same loss of dextran fluorescence over time. And so by using our established method of micropuncture, we studied the effects of alveolar type 1 cell CFTR deficiency on alveolar wall liquid secretion. So this first image here I'm showing is an alveolar field from a litter-mate control mouse. The fluorescent epithelium is digitally removed so that we can just focus on the dextran fluorescence in the alveolar airspace. We imaged the same area after 15 minutes and 30 minutes, and you can see that the tracer fluorescence is lost over time, suggesting that the tracer is being diluted by secretion of non-fluorescent alveolar wall liquid. We also quantify the group data here, and this downward bar shows the percent loss of dextran fluorescence from initial. This next image here is similar, but is from an alveolar type 1 CFTL null mouse. We again image the same area after 15 and 30 minutes, and you can see that the tracer fluorescence loss is at a much slower rate, suggesting that the alveolar wall liquid is reduced, which is also what we see with the group data quantifications. Now, interestingly, when we looked at the group data for the alveolar type 1 CFTL null mouse, we saw this splay of dextran fluorescence loss within that same group. So we went back and looked at the Cree expression levels in these alveolar type 1 CFTL null mice. And we noticed that within the same lung, there were areas of low Cree expression, medium levels of Cree expression, and high levels of Cree expression. The black areas that you see here, they have alveoli, but with magenta fluorescence that I'm not showing you, so that we can better appreciate the different levels of Cree expression or cyan fluorescence. This heterogeneity of cyan fluorescence has been reported by others, so I won't get into too much details about it for today's talk. But we actually wanted to use this heterogeneity as our advantage for our studies because we can determine liquid dynamic in regions with more or less Cree expression within that same lung. So for each of the Cree expression levels, we again looked at dextran fluorescence in the alveolar air spaces. In areas with low levels of Cree expression, we again see rapid loss after 30 minutes, whereas in areas with medium level or high levels of Cree expression, we see that the dextran fluorescence loss becomes more gradual, suggesting that the level of CFTR deficiency relates to loss of dextran over time. We quantified the dextran fluorescence for each Cree expression level, and these are the group data here. I'm also going to show you the same data, but plotted as a scatter plot, where we plotted the Cree expression level in the alveolar type 1 cells on the x-axis and the dextran fluorescence loss on the y-axis. By doing this, we interpret that there is a linear relationship between the extent of CFTR expression in alveolar type 1 cells and the extent of alveolar wall liquid secretion. These data, combined with our imaging data, we conclude that CFTR expression in alveolar type 1 cells contribute to alveolar wall liquid secretion in a dose-dependent manner. So with these new understanding, we next wanted to look at the physiological relevance of these of these results on how CFTR expression, specifically in alveolar type 1 cells, shape alveolar defense, considering that alveolar wall liquid secretion protect alveoli from inhaled pathogens. So we asked, well, do CFTR in alveolar type 1 cells contribute to bacterial clearance? To do this, we intranasally instilled the mice that were either littermate control or alveolar type 1 CFTL null with staph bacteria. We then viewed the lungs under confocal microscope, as I've described earlier. These mice in these staph experiments, they don't have the MTMG allele because our staph bacteria are GFP tagged. So these first sets of images are showing an alveolar field as you've seen before, but with the alveoli labeled by calcine dye so that we could delineate the alveolar epithelium. You can also see that the distribution of the staph bacteria is similar between both mouse groups. To better focus on the staph clusters here, these next images are showing just the cyan fluorescence of staph bacteria 1 hour after its intranasal installation. This next image in the control mouse lung was taken 3 hours later in the same area, and you can see that the staph bacteria have largely disappeared from the alveolar air spaces. But if we look at the staph clusters in the alveolar type 1 CFTL null mice, you'll notice that the staph have remained steady over time. So we quantified the staph data here, and then combined with our imaging data, we conclude that alveolar type 1 cell CFTR contributes to staph bacterial clearance in alveoli. Finally, to summarize, we found that CFTR in alveolar type 1 cells contribute to alveolar wall liquid secretion and shaped alveolar defense by clearing inhaled staph bacteria from the alveolar airspace. A major theme that our data highlights is that alveolar type 1 cell CFTR is important for alveolar homeostasis and defense. And since the alveolar epithelium comprises over 95% of the lung surface area, we suggest that developing therapies for CFTR-related diseases should consider alveolar type 1 cells as a therapeutic target. Thank you all for your time, and I'm happy to take any questions that you may have.
We take questions maybe while people come up to ask. I ask the first one. So Wanda O'Neill brought up the high expression of CFTR in 82 cells, and you were carefully to say they have CFTR function and 81 cells contribute. What would you expect if you would do the same experiment, knocking out CFTR in 82 cells? And what, in your opinion, is the shared function of CFTR in 81 versus 82?
Sure. Thank you for that question. Yeah, so I didn't have time to add those data in my talk today, but we did. We have the same exact breeding program where we also did CFTR null expression in alveolar type 2 cells, where we took an SPC specific mouse. And we also showed that both alveolar type 1 and alveolar type 2 cells do contribute to alveolar wall liquid secretion. And we have that data actually in a bioarchive paper, but I couldn't include it in today's talk, of course. And your second question in terms of differences of CFTR function in type 1 or type 2 cells, we think that both cells contribute to alveolar wall liquid secretion, but it's likely that CFTR expression in both cell types are different. But we think that we've also have data where we knocked out CFTR in both cell types together in the same mouse, and we saw that there was basically no secretion in when we knocked out CFTR in both.
Thank you.
Yeah. Thank you.
Those are very cool experiments. Congratulations.
Thank you.
But I have a question that's going to reflect my ignorance. Does the Cree-induced expression of the GFP imply necessarily that CFTR is expressed?
So thank you for your question. So to repeat that, you want to know if the Cree expression directly relates to CFTR deletion or deficiency?
Well, so the CFTR is floxed and you would knock it out, but if you knock it out and its expression is never being driven for whatever reason, but I don't know your construct, but your GFP would get expressed.
Yes, exactly. So we have our GFP allele right after the CFTR flox. So once Cree, snips away the CFTR flox sites, then it will express GFP. And we've also done qPCR experiments with our alveolar type 2 cells to show that the alveolar type 2 cells that we pulled out that are GFP positive, they do not have CFTR mRNA.
Okay.
Lovely work, cannulating a mouse lung. That's very impressive.
That's all of our lab
Yeah,
technique.
lovely. It's really lovely work. But I understand, though, it's a balance between secretion and absorption, all right, throughout the airways, especially in the alveolar sacs. So did you consider the role of CFTR potentially in absorption, since we're debating backwards and forwards of ionocytes and their role in perhaps both functions? CFTR is playing a role in both.
Yeah.
Is it possible here, and did you even look or consider looking at the role of sodium? Because I understand that's important in absorption as well. So,
yeah, thank you for your question. Yes. So kind of a long way to say this, but there are published data that has shown that in the lungs, there are evidence of secretion and absorption. For our studies, we are doing more experiments and control experiments to really determine which way it's going, secretion or absorption. But I can say that in our previous studies with alveolar wall liquid secretion, we've shown that in our model, there is baseline secretion when CFTR is intact. And when the lungs are flu infected, actually, we see that there's absorption, and there might be a relationship between CFTR and ENAC channel, where once CFTR is inhibited, it looks like the ENAC channel gets turned on to induce some sort of absorption. Now, the mechanisms need to be more elucidated, and that's what our future studies will look into. But yes, we are trying to sort of look into more studies, control data to see whether the lungs are secretory or absorptive.
Hi, Viral Shah, MGH. I just wanted to echo what the other questioners asked. Really cool prep. It seems very hard to do. So great job there. I probably missed it at the end. So you saw less bacteria when CFTR was, more bacteria when CFTR was knocked out. Was that right? Or was
Yeah,
it the other way around?
it remains steady over time after 3 hours of installation.
My question was, I know we're thinking about like fluid secretion absorption. Could it be some other mechanism that's involved? Could it be like antimicrobial killing, or did you look up to see in the more proximal airways? Do you see more of that bacteria cleared versus not to help with understanding the mechanism?
Sure. So to, I guess, address the last part. So with our imaging technique, we can't actually see the airways. We're imaging just the subflora of the lungs.
Very distally, right by the pleura.
Exactly.
Gotcha.
So when we look at, when we're imaging about half an hour just to look at the liquid dynamic. Mm-hmm. And during that time, we don't think the immune system could contribute. I mean, it could be, but we're not focusing on the immune cells. And but in our previous data, in our previous publication, we've also shown that the immune cells, when the staff are present, don't come in until maybe 9 hours, like later in the
I was thinking more about the innate defense of the epithelium itself.
Right.
Could be contributing there.
Yeah, certainly. As you can see, that even in our littermate control mice, not all of the staff disappear from the alveolar aerospace. So there's certainly other mechanisms at play here. We're just adding to literature that alveolar type 1 cell CFTR function help with bacterial clearance through alveolar wall secretion.
And your trachea is cannulated, right?
Trachea is cannulated.
I wonder if you could use that to your advantage and just collect all the secretions through the cannula and say, oh, this is how many GFP bugs we had in one condition versus the other, to try to help with your clearance.
Yeah, that's tough to say because we have to obviously keep the lungs and the trachea intact so we can connect it to our microscope system. We've done BAL with through the trachea, but that's
just something to think about.
Yeah.
Cool. Thanks.
Thank you.
Oh, thank you.
All right, I want to Say an honorary thank you to Gary Cutting for the setup question for our next talk, where we'll move more proximally and start thinking about CFTR function in ionocytes. Really excited to have Jay Luan here from University of Saskatchewan to share some insights into the ionocytes. Thanks, Jay.
So good morning, everyone. Thank you for the chair and the organizer for giving us the opportunity to present today. And I have no relationship to disclose. So since the discovery of ionocyte, it's become one of the hottest topics in the cell field since the ionocyte express very high level of CFTR messenger RNA. However, it's a really rare cell type. Only count about 1% of cell in the airway epithelium. And it has been proposed that abnormal function of ionocyte contribute to the development of CF lung disease. And however, there's still debate about how the loss of preliminary ionocyte function lead to CF lung pathogenesis. So in my project, I'm focused on the distal airway because it has been shown that CF lung disease start from the distal airway, and in the distal airway in the tissue, also the distal airway have a very unique structure. It has folds and the pleat of the distal airway. And evidence suggests that there are different groups of cells at fold and at pleat. And the CF lung shows that there's an abnormal airway surface liquid there. And this abnormal ASL contribute to the CF lung disease. To protect the airway, it requires a healthy ASL there. And the mucin secrete there require unpacking. And to unpacking those mucin, you need appropriate pH and bicarbonate concentration in the ASL for the helping for the process. So we hypothesize that the ionocyte secrete bicarbonate actually facilitate the unpacking of the mucin in the distal airway. And this unpacking process requires for the mucociliary clearance process in the distal airway, which protect the airway. And in my project, I have two goals. One is to figure out the location where ionocyte locate and those mucin secreting cell locate in the swine distal airway. And then we want to describe the function of ionocyte to ASL formation in the distal airway. So first, we want to see where the ionocyte locate, and also those mucin secreting epithelial cell locate in the distal airway of swine. So we use paraffin embedded swine distal airway. We dissect from fresh harvested lung, and we slice the paraffin embedded tissue. And here is swine distal airway slice. The airway is about 1 millimeter diameter in this case. And we stain the different cell type, use BSND for preliminary ionocyte. We also staining the mucin 5AC plus cell and mucin 5B plus cell. And you can see the fold and pleat of the distal airway structure. And here is a zoom in picture about one section of the swine distal airway. And we can see most of the airway surface actually covered with ciliate cell. We can see the cilia here. And then we can see the BSND labeled cell there, and we can see mucin 5B plus cell and mucin 5AC plus cell. And there's some mucin 5AC plus and mucin 5B plus cell in the same cell. And then we moving forward to quantify where those cells locate. So we set the tip of the fold of a distal airway as 100% distance from the pleat, and 0% is the very bottom of the pleat of the swine distal airway. And as you can see, we quantify the ionocyte. It's actually ionocyte present from the fold to the pleat all the way. However, most of the ionocyte actually locate at the fold region, about 50% distance from the pleat. About 80% of preliminary ionocyte in the swine distal airway is at the fold region, and only 20% in the pleat, 80% in the fold region, and only 20% in the pleat region. And about more than 55% actually at the very top of the fold region of the swine distal airway. And the mucin 5AC cell is distributed differently. It's kind of evenly distributed from the fold to the pleat. And mucin 5B plus cell, actually most of them locate at the pleat region. So we conclude that preliminary ionocyte in swine most appear at the fold region of the distal airway. And then we move to describe the function of ionocyte in the swine distal airway. So here we use a technique called self-referencing ion selective microelectrode technique. It's a technique with super space and time resolution. We can measure ion transport through a single cell, and we can record the ion flux in real time. So here is how the technique works. So if an epithelial cell consistently moving ion from one side to another, the constitutive flux generate ion concentration different in an ancestral layer close to the tissue. And we can use that microelectrode to pick up the gradient difference close to the cell, and we can calculate back how fast the ion move through that single cell. So we collect the distal airway tissue, smaller 2 millimeter in diameter, from the swine lung, and we incubate with metal tracker. And we use a metal tracker to screen those mitochondria-rich cells. Since preliminary ionocyte is one mitochondria-rich cell, it's highlighted in those metal tracker scanning imaging. And then we move our electrode to that cell, and we start measuring the ion transport property there. And after we've done the experiment, we fix the tissue, and we use BSND antibody to confirm the cell that we measure is preliminary ionocyte. So metal tracker highlights the mitochondria in the cytoplasma in the ionocyte, and the BSND antibody label the basolateral memory of the preliminary ionocyte. So here's a result. We measure whether preliminary ionocyte affect the pH or chloride or sodium movement across that preliminary ionocyte. So and here we show that the y-axis is how fast the ion move, and the x-axis is the time. We add first choline IBMX at 150 seconds. And if we see a positive flux number, it means the ion is moving to the lumen. And if we see a negative number, it means the ion is moving to the basolateral site. So after first choline IBMX stimulation, the ionocyte shows that there's a alkalization of the airway lumen after first choline IBMX. And the chloride ion actually moving from the luminal side to the basolateral side. And even before and after first choline IBMX stimulation, there's almost no sodium movement through that single preliminary ionocyte. We also measure some the ion transport property from those club cells in the distal airway. It shows a totally different story. So after first choline IBMX stimulation, there's a huge chloride and sodium transport from the basolateral side to the lumen side. And there's a little movement of pH change through the club cell. So this indicates that the sodium and the chloride ion actually moving from the basolateral side to the apical side through club cell, and the sodium and chlorine movement actually is a driving force for the liquid secretion there. So we think that the club cell actually regulate the ASL volume in the distal airway. Then we wonder what happens if we block CFTR channel there. So we use GlyH to block CFTR channel in the swine distal airway tissue. And if we incubate the tissue with GlyH, actually it blocks the pH modulation through ionocyte. So, and we quantify this change to calculate the area under the curve. And it shows that
A little bit more lights, please.
it shows that it's blocking the pH modulation there when we calculate the area under the curve after forskolin IBMX stimulation. And then we're wondering how the ionocyte modify the pH in the ASL. So one way is by secreting bicarbonate. So what we do is we use acetozolamide to block the carbonic anhydrase activity inside the pulmonary ionocyte. Also, we remove the bicarbonate ion in the bath solution. And removing all the bicarbonate ion there actually block the activity of ionocyte. And we quantified it with area under the curve. So it means that our result indicate that the ionocyte actually secrete bicarbonate to modify the ASL pH. And then we wonder how the bicarbonate secrete. So our previous paper show that actually pulmonary ionocyte express AE2 ion exchanger 2, and DIDS blocked ion exchanger 2 channel, which blocked the bicarbonate going into the lumen of the airway. And the DIDS has shown that not block the CFTR channel. We confirm that use the club cell, and also we using chamber for our distal airway sample show that DIDS do not block CFTR activity. However, DIDS here block the pH modulation stimulated by forskolin IBMX through the ionocyte. So we think that pulmonary ionocyte play a role in bicarbonate secretion in the distal airway. So DIDS, we think DIDS is a tool that are blocking ionocyte function in the swine distal airway. Also, we study other stimulate, such as UTP. UTP is very interesting because Dr. Okuda in 2021 in the Blue Journal showed that actually pulmonary ionocyte do not express the P2Y receptor, which sensing the UTP, extracellular UTP. So here we show that actually UTP do not stimulate ionocyte function in the swine distal airway, and actually unlike what we got from forskolin IBMX. However, UTP do stimulate the club cell function at the swine distal airway, and it show very similar response compared to forskolin IBMX stimulation. So we think that we can use UTP as a tool that stimulates the secondary cell there, but not ionocyte. So then we move on a functional study of the distal airway. So what we do is we collect airway surface liquid from distal airway. We loaded those ASL with FITC dextran, and we photobleach the region of the ASL. And after the photobleaching, there's a recovery period because the mucin try to go back to the place where it have been bleached out. And this recurring process indicates how viscous of the ASL. Here is the recovering curve. So we collect the ASL from forskolin IBMX stimulation. The half time of the recovery is about 5 seconds. However, if we block ionocyte function with DIDS, there's an increase of recovering time. So this indicates that the ASL collect from that condition is more viscous. However, if we put back the bicarbonate ion into the ASL we collect, actually it's recovery those increased viscosity. And the ASL collect from UTP stimulation have a higher recovery time, indicate a higher viscosity there. So the ASL, so in the UTP condition, if we stimulate the secondary cell but not ionocyte, there's an increase of viscosity. So in summary, we think preliminary ionocyte clustered the fold of the distal airway, and our microelectro experiments show that ionocyte play a role in bicarbonate secretion in the distal airway. So we hypothesize that the bicarbonate secrete through ionocyte actually affect those mucin secrete from mucin 5AC and mucin 5B cell. And when the mucin goes through the ionocyte, the bicarbonate secreted by ionocyte help the unpacking process. And the club cell seem to contribute to ASL volume. And we also want to investigate the role of ciliate cell in ASL formation. Our preliminary data actually suggests that the ciliate cell also play a role in the ASL formation in the distal airway. So finally, I would like to thank my supervisor, Juan Asiki, Julian Tam, and Veronica Capnucci, and all the lab members involved in this project, and all the funding agency to support our work. Thank you.
We'll open for questions for Dr. Lan, and while he answers questions, I'll ask everyone's forgiveness while I go back and fiddle with the lights.
Yeah. So I assume that the buffer system was CO2 bicarbonate. In your model, you had lots of CO2 supplying the bicarbonate, right?
Yes.
Yeah. Why did you put the chloride bicarbonate exchanger on the apical side? I think your data would be equally consistent unless you have some evidence from the sidedness of the DIDS effect with an exchanger on the basolateral side.
Actually, we have immunofluorescent data show that the AE2 channel is expressed in the preliminary ionocyte there. Yeah.
In the apical membrane?
In the apical memory, yeah.
Watch out.
Sorry.
Excellent talk. I wonder,
Just ignore the lights for now.
should I stop? So you said ionocyte function is sensitive to these, but how about CFTR 172? Have you tried to use?
Oh, we tried CFTR 172. It's also block the, yeah.
AJ, great talk as usual. So, when you read about Airways, it's a sodium absorbing epithelia. And I was just wondering, do you think there's a little bit of sodium absorption coming from every cell, or have you not just found the golden ENAC cell yet?
From our experiment, actually, we try amiloride, and it didn't block, it didn't show any effect on preliminary ionocyte and club cell. So we are wondering whether it's from ciliate cell, and we are working on it. Yeah.
Thanks.
Thanks again. Great presentation. Oh, Wanda, sorry.
Well, I just think this is super interesting. And, I always thought the pleats and the folds, like the distal airway expands when you breathe, they go away, and then they come back during this fixation process. And it was kind of always like an artifact of collapse during collection. And then the fact that, so do you think this is only active when we're not breathing? And how do you think about pleat versus fold in this process?
Yeah, Dr. Paul Quinton actually published a paper a long time ago, talk about that process, and he called this washing machine effect. So it's really interesting. We're wondering, what happened? We don't have live model to see what's happening, but it would be interesting to see actually how that washing machine effect happening and how that works.
Wonderful. If there are no further questions, we will move on to the last presentation.
So we're going to finalize our proximal march by Dr. Ma, coming to us from University of Alabama at Birmingham. And we're going to think a little bit about the submucosal glands and myoepithelial cells. Thanks, Dr. Ma.
Okay, thank you, Cheers, for your introduction. Good morning, everyone. I'm Yabo Ma from the John Engelhardt Lab at UAB. Today, I will present my work on ferret airway glandular myoepithelial cells exhibit regenerative potential with enhanced CFTR function of progeny epithelium. I have nothing to disclose. Cystic fibrosis is a lethal autosomal recessive genetic disease caused by CFTR mutation that disrupt chloride and water transport, leading to thin mucus infection and lung damage. Airway submucosal gland, also called SMG, they are the major source of airway mucus. Located in the cartilage airway, they secrete antibacterial factors, mucus, and electrolyte fluid to maintain homeostasis. Importantly, airway submucosal gland have been demonstrated as a predominant set of CFTR expression. The abundant CFTR is more important for airway homeostasis. However, in CFTR patients, these glands showed severe hypertrophy and hyperplasia, suggesting an aberrant proliferation and differentiation of glandular stem cell may undergo along with the pathogenesis of CF disease. We can see in here, airway submucosal glands are composed of cellular cells, mucous cells, and myoepithelial cells. In addition to their secretory function, recent studies showed that airway submucosal glands also serve as protective niche for airway stem cells. They provide localized environmental signals to maintain and mobilize these cells during turnover and airway injury. Here, nanotreating mice revealed that a subset of glandular myoepithelial cells identified as resident stem cell, they were able to regenerate ciliated cells, mucous cells, and club cells of surface airway epithelia following severe injury. Because myoepithelial cells express alpha small muscle actin, which is encoded by ACT2 gene, so our lab in mice used ACT2 Cre-RT2 RUSA reporter system to nanotrace myoepithelial cells. After tamoxifen induction, we can see the myoepithelial cells surrounding ICA9 were labeled with GFP. Following severe injury, this cell migrated the surface airway, so we can check their progeny by detecting the GFP cells. However, in mice, some mucosal glands are sparse and limited to proximal trachea, and the airway epithelial cell types is different from the humans. We can see here club cells are major secretory cell in mice airway, and ionocyte cells that CFTR-rich cells type expressed in proximal trachea. While in the humans, pigs, and ferrets, the abundant submucosal glands throughout the cartilage airway. Here, goblet cells and club cells are major secretory cells for their airway, and ionocyte expressed in the cartilage airway, so suggesting inferior lung biology, including cellular composition and structures, is more similar to humans. And here, pathologically, In CF, adult pigs and ferret showed phenotypes that mucus plugging in the interlobar bronchial structures and abundant submucosal glands, but absent in CF mice, so suggesting ferret models are reliable model to study airway submucosal glands in CF lungs. So in our study, we choose ferret model to explore mild epsilio's contribution in CF disease. Therefore, we raise below 3 questions. First, what is the potential of SMG-derived mild epsilio's in ferret? Second, how do they contribute during airway epithelial injury and repair? And finally, do airway glandular myopsilioceal lineage have CFTR functions, and are they involved in the pathogenesis of cystic fibrosis? To answer about this question, our lab first generated ACT2 Cryrosa TJ lineage transgenic ferret model for lineage tracing, as we did in mice models. In the ACT2 lineage ferret, after tamoxifen induction, we observed the GFP-labeled cells in the bronchial submucosal gland, tracheal submucosal gland, and the extrapulmonary glands, including esophagus and salivary glands. There was colocalized with mild epsilioceal markers of small muscle actin, also called SMA. To test whether mild epsilioceals involved in the airway injury and repair, like 2 mice, we gave the tamoxifen and to ACT2 lineage ferret and then injuried with intratracheal spray of polydognol, a detergent. 2 weeks later, we can see a large cluster GFP positive cell as shown on the left epifluorescence images of the whole mount tracheal lumen surface. In red panels, we can see GFP-labeled mild epithelial cells covering the tracheal and the bronchial surface and repopulate the airway epithelial cells. Here, we can see these cells can migrate to the basal layer and filling within the gland. To identify lineage cell types, we colocalized the GFP with airway epithelial cell markers like FOXG1, KRT5, and SJB1A1, suggesting the mild epithelial lineage cell were differentiated into ciliated cell marked by FOXG1, basal cells marked by KRT5, and secretory cells marked by SJB1A1. In here, to further analyze the proliferation and differentiation potential of mild epithelial cells, we first isolated GFP-labeled glandular mild epithelial cells by backsorting and 2 collected GFP-labeled MEC fraction and TD tomato-labeled no-MEC fraction. After 21 days of differentiation, we can see the GFP-labeled mild epithelial lineage cell can give rise to ciliated cell marked by FOXG1, basal cells marked by KRT5, goblet cells marked by MAC5AC, club cells marked by SJB1A1, and ionocytes marked by BSND. Next, we want to know whether mild epithelial lineage cell retain CFTR response. So, see as above, isolation and AR culture, we used using chamber assays to measure short circuit current to reflect CFTR response. In here, we observed in the early passage, MEC lineage cells have a ISC increase after IBMX phosphocline stimulation and inhibitor GLH1L1 almost abolish the IBMX follicle-induced current. This confirms CFTR-dependent response, but it's lower compared to non-MEC fraction. In contrast, in the later passage, we can see the MEC lineage cells have a higher CFTR response compared to non-MEC fraction. In here, our quantification result showed in the later passage, we can see the MEC lineage cell have a higher CFTR response compared to non-MEC fraction. In right graph, we can see CFTR activities of myelapsylin lineage cell was enhanced as the cells were passaged. Next, we asked whether myelapsylin cells involving airway submucosal gland remodeling during CF disease progression. So in Act 2, lineage trace the CF ferret were started to develop CF disease by withdrawing VX770 at the age of 3 months and tamoxifen labeling at 4 months and evaluated at 14 months. In here, we didn't observe their proliferation into the surface airway apsili cells like severe injury. Instead, we observed the new regenerated glandular IC9 cells and the lineage mucus cells and serous cells by immunostaining. This data imply myelapsylin lineage cell involving airway glandular hyperplasia and remodeling during CF disease progression. In summary, we found myelapsylin cells differentiate into basal cells, secretory cells, and ciliated cells during airway repair. And in vitro myelapsylin lineage cell can give rise to multiple airway apsili cells that retain CFTR response. And in contrast, CF glandular myelapsylin cells contribute to submucosal glands remodeling. Finally, I want to thank everyone in Engelhardt Lab, of course, our ferret for doing this part. Thank you all for your attention. With that, I will be happy to take any question.
We'll go ahead and open to questions for Dr. Ma. While people come up, I'll start with the first one. What's your hypothesis for why the CFTR activity is increasing over passage in those myeloepithelial subglandular cells?
Are you asking hypothesis in the CFTR?
Why is it increasing with passage? Is it a change in expression? Are the cells differentiating differently? What drives that?
In CF, because CF is the CF disease is a CFTR mutation, so airway submucosal structures have abundant mucus and less water secretion, so make some gland structures hypertrophy and hyperplasia. They can, like this. Thank you.
I think it was also the question, why is CFTR function increasing?
Yeah.
Yeah, because of that.
In CFTR, in the early passage, we can say myelapsylin cells derived apsili cells have the CFTR response, but it's lower. You can refer to Tata's picture. In the early passage, we can see the myelapsylin cells transition from ACT2 positive cell to ACT2 negative cells and change to the basal cells. So maybe in the later passage, the basal cells have the robust potential to differentiate some multiple areas apsili cells.
Thank you.
Thank you.
Beautiful presentation, Yabo. And I don't know if you mentioned this, but from what I know, myoepithelial cell marker as SMA is not specific, right? So how were you able to ascertain that the regeneration is from myoepithelial cells?
Thank you. Your question. Yeah, you are right. You are right. Small muscle actin encoded by ACT2 is not specific markers. They also label the other like vascular small muscle, airway small muscle, even myofibroblast. But you also can refer to Tata's paper because myoepithelial not only marker alpha small muscle actin, they also express the K5. So in Tata's paper, we can see in ACT2 lineage mice models, myoepithelial cells can migrate to the surface airway with co-staining the KRT5 and the KRT8. So I think these cells derived from myoepithelial cells. Thank you.
Great talk, Yabo. Thank you. I'll ask you a question that I always get asked when I present anything about glandular myoepithelial cells is, have you thought about if glandular myoepithelial cells in different glands, in nasal tissue versus trachea versus interlobar airways, or maybe even across species, mouse tracheal glandular myoepithelial cells versus, and do they, like your assay where you are able to sort the GFP expressing MECs and then differentiate them in ALI, do they have the same potential? Are they exactly the same? Can you put a distal, quote unquote, distal glandular myoepithelial cell in a proximal airway? Does it function the same?
Yeah, that's a good question. Some mucosal glands throughout in the cartilage airway, so in the tracheal, bronchi, interlobar bronchi, and some have abundant some mucosal gland. I don't know myoepithelial cell potential of tracheal or bronchi is different, but it's a good question. Maybe I can investigate further. Yeah, thank you.
Thank you so much for a great talk and also making me realize ferrets are such so cute animals because I always see dissect tissue and the histology. So yeah, so I have one quick question regarding stem cell properties of myoepithelial cells. Because in human lungs, there is no submucosal glands in the distal airways. So that might be one of the reasons, because that means small airways lack stem cell reservoir. So then that could be one of the reasons why small airway regions are a little bit vulnerable to lung injury. So, but how distal can these myoepithelial cells go to the distal lungs if lungs are damaged? So that, they can repair all epithelium to the very distal part, or there is some limitation? If you have any insight into that, so that'd be great.
Yeah, that's a good question. We focus on the myoepithelial migrated airway epithelium to repopulate anything. In the distal lung, maybe myoepithelium involve this progression, but I don't know. Sorry.
Yeah. Thank you.
Okay, one last question.
Hi, very beautiful model. So I'm wondering whether you've looked at whether this type of repair mechanism from this source of stem cells may be contributing to some form of maladaptive repair. So given that there is this large-scale injury component, I'm wondering if you've looked at maybe the emergence of squamous cell populations as opposed to normal epithelial cell types as the consequence of this type of repair from the stem cell pool. Yeah. I guess my question is is this a friend, or is this sort of, a last resort type of repair that may set the stage if it keeps happening for downstream epithelial remodeling?
You mean what some cell types will happen in the airway epithelial cells.
I guess what I'm wondering, and I'm happy to follow up with you later, I don't want to take too much time, is that if you've seen, for example, squamous cells emerging as a part of this repaired population on the airway surface. But I'm happy to check with you later. Yeah, thanks so much.
Thank you.
Thanks very much for a fantastic presentation.
Thank you so much.
I would like to thank all the speakers for their excellent presentations and the audience for great questions. Thank you for coming, and I hope you have a great rest of the day.